Tech Media Show. Today we have a special guest whose name is Freddie.
Do you have any prior knowledge on the subject restriction enzymes?
Well, to be honest, I do not, sir.
Mr. Freddie, today you are in luck because we have an expert this subject. Let me welcome to you Mr. Martinez.
Thank you. This was for having me on your biotechnology show. I heard that you wanted me to help discuss to Freddie about how restriction enzymes work.
Yes, this is true. Would you tell us an honor and giving us a brief description on how restriction enzymes work?
Well, Jesus and Freddie, the main point for the restriction enzymes is for genetic engineering. This process starts by the enzyme showing or exposing the base pairs, or as some call them, base sequences. What this enzyme likes to do is cut out phosphate or the backbone of DNA. When the enzyme does this, it is referred to as a restriction enzyme site.
Mr. Martinez, I thank you for this basic information over how the restrictions enzymes cut DNA base sequences.
Okay, can you use this enzyme to cut off DNA and place it into something else?
Yes, you can, Jesus. You can physically inject this DNA into other things or other organisms in order to create antibodies and protection against germs.
I heard that this way used in modern medicine today. Can you confirm this?
Mr. Freddie, I can confirm that this is true. It is commonly used in medicine today.
When you said that it can be injected into other organisms, what did you mean?
What I meant, Jesus, was that by injecting this into DNA or another organism, you actually help cure an infection or a small-time disease.
That sounds useful in today's society because the benefits it provides.
Indeed it will.
Okay. What are some of the components for this process to occur? Well,
some components for this process to occur is when there is a cut in the enzymes, which basically breaks it down to its basic base sequences. This cut is often the final component of the process. The last process is really up to the person controlling the experiment. The final component usually consists of injecting it into an organism in order to create a cure.
Thanks to this process, the DNA, the basic sequence are more manageable to study and easier to place into organisms?
Yeah, this is true.
Okay. Well, a lot of people and our audience ask how this procedure is used in the following people: a medical researcher, a forensic scientist, a forensic anthropologist.
Well, there is a lot of different ways each scientist can help.
What can the medical researcher do with the restriction enzyme?
Well, a medical researcher removes a specific gene from an organism and splices into another. What this means is he physically cuts the DNA and injects it into another organism in order to create cures for cancer, such as oncology and other things.
Well, what about a forensic scientist?
A forensic scientist uses this method in dealing with blood samples in specific crime scenes. This is done in order to catch the bad guy by using his own DNA as evidence.
A forensic anthropologist, what about him?
He uses the genetic analysis to determine evolutionary links between ancient and modern populations. What this means is, for example, if Freddie was my ancestor, we could trace my blood back to him using restriction enzymes.
Well, that sounds really great, but now it's time to go. And thank you, Freddie, for coming.
On the show.
For
Thank
on
you
the
very
show.
much.
Thank you, Mr. Martinez.
Welcome anytime.
Thank you. And well, we have to go now.
Thank you for coming to the
Mm,
Medical
Medical
Biotech
Biotech
Show.
Signing off.